I think the biggest thing for me in the cell therapy space that's the biggest challenge is really just trying to balance the reproducibility from a manufacturing perspective as it relates to the variability that we're seeing from the patient material. And so that's one of the biggest challenges that we have, trying to understand and balance the product quality aspects, the patient safety aspects, and consistency aspects, and how that relates to where you would typically move that through in a typical biologic, and how does that relate to the cell therapy space where you have this individualized medicine really based on that patient input material. Because it's so highly variable, it is very challenging for us to try to understand what's the right space and what's the right controls to have in place. The other piece is really trying to work with the agencies to understand how flexible can they be given this space? Where are we able to expand and be more flexible compared to a typical biologic? And I think that's one of the key things that I think we're trying to strategize to see where we can go at Juno and where we can push the envelope on things and work with the agency on those kind of things. Getting back to the big challenge, I think, is just trying to find that right balance of manufacturability, obviously reduce costs, patient access, and then working with the agencies to make sure that they understand where we're coming from so we can have a control strategy that's suitable, that's wide enough to support the dramatic patient variability that we typically have, but also controlled enough to meet the agency needs for those type of controls. The biggest thing that we can do is really work with the agencies and really educate them and trying to understand how we are building our portfolio, our framework for the manufacturing, and really trying to have them understand where we're coming from. I think the other piece that we're really trying to work towards is to really bind the critical quality attributes to the actual clinical outcomes through a very unique correlative analysis. And really for us, what that means is we can really go to the agency and really say, look, here is a broad range of data that supports this particular quality attribute, and it's okay. And from a safety and efficacy perspective, we can show that it is not adversely impacted across that entire range. And so working with the agency to help them understand that, and from that concept, it really helps build, the future state from where we'd like to be. From our perspective, what we'd like to do is really, through our process characterization, through our product understanding, through the many number of batches that we're manufacturing to support patients in these clinical trials, really trying to drive, just like we had indicated, through that correlative analysis to understand the variability that we're seeing. Is it practically important based on the clinical outcomes? And so if you look at the clinical safety and efficacy claims that we're trying to build into our clinical trials and ultimately to the BLA in the future, working with the agency to make them truly understand that the data that we have and that we're collecting is really able to support those broad claims. And so I think having them understand all of those pieces is critical for us as we move forward in the future, really engaging the agencies and really trying to help them understand where we're coming from.